Okay,
Okay.
then let's get going.
All right.
So thanks to everybody that has logged in online. I'm Beth Workett. I am one of the senior scientists with C-DEBI, and it's my great pleasure to introduce today's talk. Before I introduce Drew, I just want to remind folks who are on that this is part of the network speaker series that C-DEBI puts on roughly every 2 months or so, although we're going to take a little break through the summertime when most folks are busy with field work. So our next network speaker series will be on September 7th, featuring Gustavo Ramirez, who is a postdoc at the University of Rhode Island with Steve Daunt. So please mark your calendars for September 7th. And also to let everybody know that next month, June 15th, which is a Thursday, there will be a professional development webinar hosted by C-DEBI that will be on the topic of preparing proposal budgets. And this is meant for postdocs and graduate students, anybody who wants professional development skills, to learn kind of the ins and outs of getting started with writing proposals, proposal budgets, I should say. So that's on June 15th. There'll be an announcement about that going out soon. But again, thank you to everybody for logging in for today's talk by Professor Drew Steen from the University of Tennessee, Knoxville. Drew got his PhD and his master's from UNC Chapel Hill, working with some of great folks learning about organic carbon and how to study it in the environment. I first got to know about Drew's passion about studying this kind of stuff when we were both graduate students participating in the geobiology course that was hosted at USC way back in the day. And so I'm really excited to hear about what Drew's been up to since starting at Tennessee. So thank you, Drew, for joining us today.
Thank you very much for having me, and thanks everybody for listening in. And feel free to, if you want to, all the people listening, feel free to break in at any time by raising your hands in the Adobe Connect software, and I think Matt will let me know that you have a question. So before I get started, I just want to mention that my lab is recruiting PhD students here in Knoxville, Tennessee. This is a pretty nice place to live, I think, and we're doing some interesting science, I like to think also, although I guess you can be the judge of that over the next half hour or so. So I want to start out by talking a little bit about what it means to talk about recalcitrant organic matter. So a lot of my work on subsurface sediments, I'll have some kind of sentence in an introduction that says something to the effect that it's surprising that a lot of the microorganisms in subsurface sediments appear to be heterotrophs because the organic matter in those environments is really recalcitrant. And in one sense, that's an extremely straightforward thing to say. The organic matter that is in those environments has been there for a really long time and it hasn't gone away. But I actually think that that term recalcitrant can also tempt us into thinking about it in a somewhat misleading way. So the most useful way to specifically talk about reactivity of organic matter or recalcitrance is with geochemical models. Like, for instance, on the left here, I have these equations from Westrich and Berner's multi-G model, which posits organic carbon as the sum of independent pools of organic matter, each of which has a different reactivity. That is to say, it decays with a different rate constant or half-life. So you divide the organic matter in the environment up into some number of pools and watch it go away with time or with depth in the sediment or whatever. And that tells you about the quantity of different pools of organic matter. And there's all kinds of variations on those models. So Jack Middleberg introduced a similar model like that, where the reactivity rate constant is a function of time. And Doug Leroux and Hans Roy have had a lot of successes, as well as some other people lately, with what they call the reactivity continuum model, which posits an infinite number of pools of organic carbon, each of which decays with a different rate constant. But what these models have in mind, or have in common rather, is that they put us in a frame of mind that there are these different sets of organic carbon. They have a reactivity that is set by whatever it is set by, and thus it ever was. Amen. And we know that that's not really true, right? We know that there's lots of cases in which organic matter reactivity is actually really contingent. So I'm just going to show you a couple of relevant examples of these, although just before this talk, I kept coming up with more and more examples that I thought, oh, I should throw that in. But on the left, there's this, this image from this paper by Katrina Edwards and colleagues from 2001, in which they grew microorganisms on a polished sheet of shale from Kentucky. And the point of this is just that the microbes sort of aggregated around spots where this ancient organic matter was enriched. They're clearly growing off the ancient organic matter, which would appear to be incredibly recalcitrant, and yet is able to support a relatively fast-growing group of microorganisms. The figure on the right is a little bit more subtle. It's from a paper by Carol Arnosti and Nico Fink and colleagues, from 2005, not 2001, like I wrote in GCA. So this was a paper about differences in reactivity of extracts of organic matter from different species of phytoplankton. And the point of the paper was sort of a little bit different, but what was interesting to me is one of the things they found is that, so this paper involved anoxic bag incubations of sediment from subsurface sediment from Svalbard. And so they, took some sediment relatively undisturbed, added some phytoplankton extract or didn't. And then at the start of the experiment, they kneaded the bags to homogenize them. And just that act of kneading the bags caused this big burst of fermentation activity and also carbon oxidation activity. So in both of these cases, there are cases where changing either, changing the physical environment changes the reactivity of organic matter. And in some sense, that's not really surprising. We know that reactivity of organic matter depends, for instance, on sorption to surfaces and on the presence of oxygen and all kinds of things like that. But the point I'm trying to make with this is just that these are cases where the reactivity of organic matter is depending on things other than the structure of the chemical structure of the organic matter. And this goes in the other direction too. This is a figure from a paper about bottle incubations of marine dissolved organic carbon. So what these authors did is used Fourier transform ion cyclotron resonance mass spectrometry, which gives us relative peak areas for something like 10,000 different distinct molecular formulae, and the relative peak area we think is something like proportional to the concentration of the molecules that form that formula. But they did a neat thing because they took these mass spectra over a time course during the incubation. They were able to estimate a half-life for each of the different peaks that they observed, and then using the molecular formula, they were able to make a guess about what class of compound that peak belongs to, and they were thereby able to talk about the reactivity of different classes of organic matter. And the only thing, I'm not totally sure if that legend is going to be visible over the webinar, but the only thing that I want to point out is that, you can see along the bottom here, there are these pie charts for the composition of the labile organic matter, the, you might say, semi-labile organic matter, and the more recalcitrant organic matter. And the only point I want to make is that those pie charts look fairly similar to each other, and that all of the compound classes are represented in all of the pie charts. That is to say, we can't have a conclusion that's along the lines of, sugars are recalcitrant or sugars are labile and unsaturated aliphatic oxygen-rich molecules are recalcitrant. Actually, all of these classes are both labile and recalcitrant. So this makes me think that if we're trying to understand reactivity of organic carbon, then we should be thinking not just about the structure of organic carbon, but we should also be thinking a little bit about what the microbial community is doing. And without wanting to get too much into microbial psychology, I think about this a little bit as like thinking about what the microbial community wants. So what does that mean? The way I like to get at this is to start with the point that heterotrophic microorganisms cannot directly take up molecules that are bigger than some size limit let's say it's something like 600 to 1,000 Daltons, because those molecules can't pass through the porins that the organisms use to take up organic carbon. And in the environment, a whole lot of molecules are a lot bigger than that size limit, and so they have to reduce the size of molecules extracellularly somehow, and so they excrete hydrolytic enzymes out, either freely out into the environment or just simply out, anchored to the outside of cell membranes, to break down big molecules outside of the cell so that they can take up low molecular weight hydrolysate. So in that sense, the set of extracellular enzymes that a microbial community is producing is in some way an index of what that microbial community wants. And I want to be clear about this, it's not an index of what the microbial community is getting, because in order to get substrate back, not only those enzymes have to produce extracellular enzymes, those enzymes actually have to go out in the world and do something. But if we can measure the activity of those enzymes, we can at least maybe learn something about what the microbes are trying to do. I say measure the activity of those enzymes. It would be nice to have a quick molecular way to get at the distribution of those enzymes, but it turns out that we really can't do it with phylogeny. So this is a figure from Amy Zimmerman's excellent paper, where she looked at distribution of extracellular enzymes in all of the cultured bacteria, in the genomes of all cultured bacteria. And these are three enzymes that are sort of key for environmental organic matter oxidation. And what you can see is that for all big taxonomic classes, there's some lineages that have those enzymes and some that don't. So we really can't use taxonomic position to very precisely predict what organic matter, what microorganisms are going to want to eat. And I'll just point out that these are cultured bacteria. It doesn't even tell us about the uncultured bacteria and the archaea, about which we know even less. And we can't actually even use gene sequences for the relevant enzymes to figure out what exactly they are doing. And the reason I say this is based off of a project that I did in collaboration with Karen Lloyd here at UT and Karolina Michalska and Andrzej Joachimiak at Argonne National Lab in the Midwest Center for Structural Biology. So the Argonne National Lab people in this collaboration were able to express and crystallize a peptidase from an uncultured archaeon that Karen had gotten a genome sequence from Aarhus Bay sediments. And the point I just want to make about this, because they had a purified sample of this, they were able to send it to me to characterize its activity. And the point I just want to make about this is that we were able to predict a substrate for this this peptidase based on sequence homology, and that was a peptide that begins with glycine proline. Using the crystal structure, the very high resolution crystal structure of this peptidase, they were able to predict that the enzyme ought to have a different substrate, D-phenylalanine. And then, using wet chemical methods, I could show that actually the enzyme had a preference for N-terminal cysteine on proteins. So these are distinctions that are, sort of in an annotating the genome kind of way. They're very fine distinctions, but in terms of, if you're trying to figure out what organic carbon is actually going to be available, those are actually pretty big, important distinctions. And of course, because we had a sample of the protein, we're also able to do things that you absolutely can't do using bioinformatic methods, like we were able to measure the pH sensitivity of this enzyme and the temperature sensitivity and all of these kinds of things. So the molecular methods are limited. We've got a little bit of progress, I think, on that front, but it's maybe not quite ready to talk about just yet. So the methods that I've been using is a much older method where we add these fluorogenic substrates. So for instance, in this example, I've got leucine aminomethylcoumarin, and that peptide bond in that molecule is subject to hydrolysis by peptidases, enzymes that break down protein. So in this case, it would release leucine and aminomethylcoumarin, which is fluorescent. And so we add some of this substrate to a sample, and we measure the increase in fluorescence of the sample over a few hours, maybe in slow environments, maybe 36 hours. And we use that to calculate the potential activity of the enzyme. That's a Vmax measurement. And like I said, this is a very old method. It was introduced first in environmental samples in 1983 by Hans-Georg Hoppe. I learned recently that it was being used for biochemistry much earlier than that, and I just have to point out that the original synthetic method for these types of molecules was to feed the precursor to rabbits and then collect the rabbit urine, in which it would the substrate would be bound to the fluorophore. So that's fun to think about. So these methods are old, but I think by applying them creatively, we can learn new things about the environment. So one example of that is a project that I got to play a small role on that was led by Nagisa Mahmoudi at Harvard. She was using an instrument built by Steve Beaupre, now at Stony Brook University, that is able to measure fluxes of carbon dioxide with very high time resolution out of aquatic samples. So she added a sample of sediment from Salt Pond in Woods Hole, or out near Woods Hole, and measured these CO2 fluxes out of it, and also collected samples to measure enzyme activity as well as a bunch of other factors, including isotopes and molecular biology, which I'm not going to address here. But one thing that I thought was neat, so the gray trace on all of these plots, and it's the same on all of the panels, is the carbon dioxide flux out of the sample, and I don't have access units for that. And then the black dots and the dark lines are enzyme activities. These first three panels are three activities of three enzymes that break down polysaccharides. Excuse me. And the last one is a peptidase. And what you can see is that there's not a particularly big peak in enzyme activity associated with this first carbon dioxide peak for any of these enzymes, and that pretty strongly suggests that at first the microbial community is using small compounds that it doesn't need extracellular enzymes to access. At some point those compounds run out, and then we see the activity of the polysaccharide hydrolases has a peak, but there's not much of a peak in the peptidase activities, relatively speaking at least, suggesting that second peak is more about oxidizing polysaccharides as well as some other compounds, as we could see from the isotopes. And then the last peak, there's again a polysaccharide hydrolase peak, but there's also a big peptidase peak, suggesting that after the polysaccharides are gone, the community is looking for for protein-like organic matter. So this is telling us a little bit about the system that would be a little bit hard to get at from more traditional molecular biology or organic geochemical methods. So that study was from a a somewhat artificial lab system in which the goal was trying to figure out what the community is doing in unlimited electron acceptors. I was interested in finding out a little bit more about what kinds of macromolecules microbial communities in subsurface sediments are trying to access. And so for that, we went to the White Oak River, and there's a preprint for this manuscript available on bioRxiv right now. It hopefully will be in print not too long from now. We went to Station H of the White Oak River, which is a really nice site because it's been studied intensively geologically, geochemically for years, going back to the 70s. It's also nice because it's a spot with really high depositional rates and high microbial activity. So there's a sulfate methane transition zone at 30 centimeters or so, depending on when you catch it, and really high rates. So you can get what you might think of as a deep subsurface environment, only 80 centimeters deep in the sediment. And so we looked at the activities of 6 different extracellular peptidases, and we focused, in that experiment, we focused on peptidases mainly for technical reasons. They're easier to measure than some of the other extracellular enzymes. And also the work with Karen Lloyd suggested that they're particularly important. And we saw, you can see in panel A, that those, the activities of those peptidases go down pretty substantially with depth, which is totally unsurprising. Cell counts and bulk activities go down with depth too. But interestingly, if we express the potential activities of those peptidases on a cell-specific basis, you can see that the activities actually don't go down all that much per cell. And these numbers of 100 to 200 attomoles of substrate per cell per hour are actually comparable to what you see in really active environments like phytoplankton blooms, surface seawater phytoplankton blooms. So what this says is that the numbers of cells are going down, but each cell apparently is producing roughly as much enzyme per cell as in really active surface environments. Mark Alperin at UNC created a model of the organic carbon oxidation rate driven by sulfate and methane pore water concentrations, and expressing the potential enzyme activity relative to that organic carbon oxidation rate shows that at depth, there's actually a lot more potential activity of enzymes per organic carbon oxidation than at the surface. And so what that says is there's about as many enzymes per cell in the deep samples, but each enzyme is returning a lot less substrate per unit time to the microbial community than at the surface. So the cells are, in some sense trying to do a similar thing at depth as to what they're trying to do at the surface, but they're doing it a little bit slower because each of their enzymes is not producing substrate quite as fast as at the surface. We also want to know a little bit more about those enzymes. So we looked at that activity of those enzymes as a function of substrate concentration, and saw first off that it fit the Michaelis-Menten curve, which is the predicted kinetics of most peptidases, quite surprisingly well to my mind. These black dots are live samples. The open triangles are autoclaved controls. So this gives us a lot of confidence that we are actually measuring enzyme activities as opposed to some kind of artifact or abiotic process. But the other interesting thing is that using these saturation curves, we can measure a binding constant for those enzymes, the Km value. And the Km tells you how efficiently the enzyme operates at low substrate concentrations. And what we see is that at depth, the Kms got smaller, which is to say the enzymes were optimized to operate well at low substrate concentrations, which is what you would expect in these deep sediments where enzymes are, where the substrate is going to be less bioavailable, or the concentrations of bioavailable substrate are lower. And so that suggests that the community is, in some sense, adapted toward to low concentrations of bioavailable organic matter. We also have some data that I just realized that I forgot to put in the talk, but I think is really interesting, that activity of peptidases that act on D stereochemistry, stereoisomer amino acids, increases relative to peptidases for L stereoisomers. So D stereoisomers are characteristic of degraded organic matter. So what this says is the deeper communities are better able to handle low concentrations of bioavailable organic matter and are better able to handle more degraded organic matter than the surface communities. And so this suggests that these deeper communities of heterotrophs are in fact adapted to a deep environment. There's been some controversy about the degree to which microbial communities can evolve or can change with depth, and I'm not weighing in on that controversy, but I am suggesting that the activities of those microbial communities, as shown by the set of peptidases that they express, does actually change with depth in a way that makes those communities more able to function effectively in the context of old, degraded, low bioavailability organic matter. But so that's that's the deep subsurface at 80 centimeters. It's also would be nice to talk about the actual deep subsurface. So my master's student Jenna Schmidt, who has now graduated, did her thesis project looking at cores from IODP Expedition 347 and the specifically the Leopoldstrate site. So she looked at the activities of some extracellular peptidases. Those are in green here. Some extracellular polysaccharide hydrolases in orange, and alkaline phosphatase in purple. And she saw down to about 50 meters below seafloor. She saw, definitely measurable activities of extracellular peptidases. The green lines here are actually a killed control, an autoclaved control. The dashed lines, somewhat confusingly, are the live sample here. And so you can see that the live sample is activities are substantially higher than the killed control. This cutoff at about 50 meters depth corresponds with an unconformity in the sediment, where below which organic carbon concentrations are a whole lot lower. And so this tells us that even in these pretty deep samples, there's plenty of extracellular enzyme activity. Interestingly, there's not a whole ton of extracellular polysaccharide hydrolase activity. Those are the orange ones. And the purple alkaline phosphatase activity, I kind of don't want to hang my hat on. It's a little bit weird. But this confirms some things that we've seen from metagenomes that suggests that the communities seem to want to eat protein more than they want to eat polysaccharides. It's not totally clear why that would be. In surface environments, we usually say that that's an indicator of nitrogen demand. In fact, a lot of these samples, there's plenty of inorganic nitrogen present, so I don't really know what that's about. But that's what the extracellular enzymes seem to be suggesting. Interestingly, Jenna was also able to look at the kinetics of these enzymes with depth, and in contrast to what we saw in the White Oak River, the binding constants of these peptidases didn't change with depth. But the binding constants were actually what we saw roughly at the deepest sediments of the White Oak River, suggesting that perhaps the enzymes are about as well adapted as they can get by our first sample, which is about 4.5 meters deep. So the last thing I want to talk about is a little bit more that we couldn't measure from molecular biology, which is the substrate specificity of these extracellular peptidases. So the substrate specificity refers to the degree to which an enzyme can catalyze reactions of multiple substrates versus one single substrate. So in, like introductory biochemistry or whatever, we're often taught that enzymes are remarkable because they only catalyze extraordinarily specific reactions. And that's actually true, except for a lot of hydrolases, which are able to accept multiple different kinds of substrate and hydrolyze those bonds. But it's really hard to know, in the environment, what those substrate specificities would be. So I'm going to spare you the detailed enzyme kinetics that I had to work out to do these measurements in environmental samples. You can read more about it in a paper from Aquatic Microbial Ecology. But what we found essentially was that, in fact, extracellular peptidases tend to be pretty what we call promiscuous. That is to say, they tend to be able to act on a pretty wide set of protein-like molecules. And furthermore, that there's a pattern to their substrate specificity. So if, on these plots, the lower numbers represents amino acids towards which the enzymes in environmental samples, these were samples from the Tennessee River and from Bogue Sound, so pelagic samples. The lower numbers are substrates to which the enzyme's presence are more active, and the higher numbers are substrates to which they are less active. And what we saw was that peptidases are more active towards amino acids which release more free energy upon oxidation to CO2. And correspondingly, they are more active towards the amino acids that are observed to be removed quickly from degraded organic matter, from organic matter during the process of early diagenesis. So this tells us that the microbial community is actually going for these substrates, these amino acids within proteins that are bigger and release more free energy upon oxidation, which makes sense. Again, microbial communities, want to get energy in the most efficient way that they can, and it seems like they're expressing a set of extracellular enzymes whose substrate specificity is tailored to produce the most energy or the most carbon per bond that is hydrolyzed. So, in conclusion, I'm sort of trying to use this talk to make the point that extracellular enzyme activities can at least give us some kind of hints as to what organic matter fractions are or aren't available to microorganisms. And in subsurface sediments, it seems like peptides are popular. Enzyme activities are detectable even in very slow working environments, like the deep subsurface, down to 50 meters or so in the Baltic Sea sediments, and that information that we get from these enzyme assays is in a way orthogonal from information that we might get from omics techniques or from geochemical measurements. And for that reason, we can use these enzyme assays to ground truth hypotheses that we generate using omics technology and using advanced organic geochemical techniques. So with that, I want to thank this whole host of colleagues in my lab and at other institutions who helped me out with this research. Particularly I want to call out Jenna Schmidt at UT, Richard Kevorkian and Jordan Bird, who are students of Karen Lloyd at UT. Also Stephen Wilhelm, who's in the microbiology department at UT, and then Nagisa Mahmoudi, Steve Beaupre, and Ann Pearson for that work on Salt Pond. And of course, my funding, a large fraction of which comes from C-DEBI and also from the Deep Carbon Observatory and from the National Science Foundation. So with that, I'm going to minimize this screen and take any questions that you all may have.
Great.
Yes. I see, interesting, I'm seeing, well, yeah, I should ask I guess if there are any questions that I can address. Okay, so Myrna is asking about an initial hypothesis. And I see that some people are typing. Yeah, so Beth asks, why do I think that omics interpretations are different from actual for the purified protein? Is it about bad reference libraries? To me, I mean, basically, yes, but that's a little bit hard on the reference libraries because very small structural details can cause a big change in the substrate specificity of peptidases. And so that's, they're not great because it's extremely hard. I should say that Jordan Bird and Brandy Reese and her group and Laura Zinke have done some transcriptomes, and I should say Jordan is Karen's student. And they're arguing, or they have some preliminary evidence to suggest that maybe they actually can predict the activities of specific extracellular peptidases from transcriptomic data alone, which would be totally awesome if that holds up. But that's very new. Those are some results that Jordan emailed me about a couple of days ago. Steve asks, how old is the sediment at 50 meters below sea level that is above and below the unconformity? And I have to say, Steve, that that information has totally left my head right now. But there is an age model for that out, and I think some of the people on this chat have access to it. So if somebody wants to pipe up with that, I'd love to hear, because I totally can't remember. So, yeah, so Myrna's pointing out that extracellular enzymes are not super labile, and they're often expressed constitutively and can be inducible. So how the community expends energy to express what it needs, and how do you explain the phosphatase? I really don't know about the phosphatase. I have somewhat low confidence that those are real data because the killed controls are a little squirrely. Understanding induction of enzymes in subsurface sediments is hard. It's hard for me to think about whether enzymes can be induced in what is really a static environment. But we don't know. Certainly, we have lots of good evidence that they're inducible in more dynamic environments, and that those induction patterns can tell us a little bit about the nature of the organic matter that's present. So we see different enzymes induced in the presence of labile proteins versus recalcitrant organic matter. But how that plays into subsurface sediments, unless those environments are more dynamic than I imagine, I don't imagine a lot of induction happening. But I'm real curious about other people's perspective on that. Maybe there's microscale variability due to viral lysis of cells or something like that. But who knows?
Cool, thanks, Drew. I think my microphone might be working now.
Yes.
Okay, yeah. So everybody, if you have questions, please feel free to enter them in the chat box, as Drew's been reading out here. And just as a reminder, in case anybody gets off before we're done with the questions, that there'll be a professional development webinar on June 15th, as well as the next speaker series in September. But please ask your questions if you have them. I'm sure Drew would love to answer them.
And Janine is pointing out that is answering my question for me, or rather Steve's question for me, that above 50 meters in the Baltic at Lillebelt is about 7,400 years before present, and there are not dates below that.
Drew, I have a question for you.
Yes, Beth.
The work you were showing, I forget whose it was, I think from White Oak, where you're trying to calculate cell-specific activity rates.
Yeah.
Vmax. Do you think it's possible to actually do it at a single cell level to look at activity?
I think it's possible to do it next week. Okay. I think I can say, without making anybody upset, that I know looks like Roland Hudson-Pickler is on this. Karen's student Joy Bongiorno was up in Roland's lab doing BondCat, so she's trying to take that technique to look at who's incorporating or who's able to synthesize new proteins in the presence of organic matter additions, and that can tell us a little bit more about what fraction of the community is able to take up complex organic matter. So that's getting towards sort of a cell-specific model. I'd be very interesting to hear anybody else's thoughts on what is possible. And Roland's saying that one can use bioorthogonal labeling to pull out active enzymes, which would be delightful.
I guess another way to frame that question, Drew.
Yeah.
So you talked about how if you look in the genomes of cultured organisms, that many organisms don't have these enzymes.
Hmm.
And so for your model, you're making an assumption of all cells. Here's the activity per cell. What do you think the variation is if not all cells are actively expressing the enzyme?
Yeah. Hashtag not all cells. I don't. I don't know. Um, I can't say
Yeah.
more than that, but I would love to know the fraction of cells that are expressing each enzyme and the taxonomic identity, or at least the distribution of taxonomic identities of those cells.
All right. Thanks. Are there other questions out there for Drew? Looks like Roland is giving some references. That's great.
I love it.
All right. And while Roland is putting in his last comments, I want to thank everybody for joining us today. If you have questions, you can find Drew on Twitter,
I
Dr.
sure
Drew
can.
Steen, and also check out his GitHub website page where you can find out all the gory details about how they do this stuff. And I want to thank you again, Drew, for doing this. It was fun to think about organics in the subsurface. And we'll see you all again soon, I'm sure.
Thanks very much to everybody.
All right, clap, clap, clap.
Goodbye, y'all.